So there are a variety of different types of mutations, and yes, we should know these types. There are nucleotide substitutions. That sounds pretty much straightforward, right? This is where you take one nucleotide and replace it with another. So if you have an A, you replace it with, say, a C, or any other nucleotide. You're just changing one nucleotide to another. Now, if this change is silent, what that means is there's no change in amino acid. This could be, shall we say, if we change GGG to say GGC, that's one codon changing to another codon just by changing this one nucleotide substitution right here. We know that both of these codons code for glycine. And so because there's no change in amino acid, it's called a silent mutation. Now, a missense mutation is where we change the DNA such that a change in one base causes a change in amino acid. For example, here's CAC, and if we change that to CAA, that would be a completely different amino acid. And so that would be a missense. You've changed the amino acid. Now, a sense mutation is where you take a termination codon, and if you remember the termination codons in DNA language, they are TAA, TAG, and TGA. If you change any of those such that it actually makes sense, it actually codes for an amino acid, that is called a sense mutation. So for instance, if we turned TAA into TGG, which is a valid codon for an amino acid, then that is a sense mutation there. The reverse of this is a non-sense mutation. This is where you take a normal codon that codes for an amino acid and turns it into a termination codon, into a stop codon. So let's say we have TAC, and if that mutates to TAG, then this, of course, means stop. And this originally means an amino acid. So this is a nonsense mutation. So we've got silent mutations, change it, but no change in amino acid. Missense, change it, and you get a change in amino acid. Sense turns a stop codon to an amino acid codon. Nonsense turns an amino acid codon to a stop codon. Here's the genetic code, and we can look at a few examples here. For instance, here's the one that we talked about. This, if you go CAC to CAA, you change it from histidine to glutamine, and so this would be a missense mutation. If you say took these two here, if we changed UGC to UGA, that would be a nonsense mutation. If, on the other hand, we take, of course, a UGA stop codon and mutate that A to a G so it becomes UGG for tryptophan, then we've made a sense mutation. We've turned a stop codon into a normal amino acid. So there's this other mutation called a frameshift mutation, and this is a little more involved. What we do is we're going to add or remove bases in non-multiples of 3 from the DNA code. And you have to think about translation here and the ribosomes reading the messenger RNA. And the ribosomes read various codons. Each codon is made of 3 letters. So here is a hypothetical codon. And here's a second codon and a third codon, and so on and so on and so on. And so we have a message in the messenger RNA that's made of groups of 3 bases, otherwise known as, in this case, 6 separate codons. And so the ribosome can read these separate words and produce the correct amino acid. Now let's say we have an insertion. Let's say we add just one base. And we're going to add a base right in here between the A and the T. We're going to add another A, so here it is, right here. Instead of the second codon reading FAT, it now reads FAA, which of course will possibly give a different amino acid. But let's look at the third codon. This codon is now completely different. And the fourth codon is completely different, as is the fifth and the sixth, and so on. So a frame shift will change all codons downstream from the mutation. It's a very, very effective way of destroying a protein. You add or remove 1 or 2 or 4 or 5 or 7 or 8 or any non-multiple of 3 bases, and it will change the whole sequence of that message. Here is the same thing but in reverse. Here we've taken away the adenine, and so now the second codon, instead of reading FAT, reads FTC, and you'll see all the codons downstream have changed as well. So these are known as frame shift mutations. Add or remove non-multiples of 3 bases. Here's a cartoon of the same thing. We can see here that here is an ATA sequence. If we change that to an AAT sequence, we see that all the amino acids downstream of this insertion have been changed. There is a kind of mutation called a trinucleotide repeat, which we alluded to earlier. And this is an example of a trinucleotide repeat. As you can see, this is trinucleotide, meaning 3 bases, right here. And this repeat just goes on and on and on and on. In this particular case, it's cytosine, guanine, and guanine. And this can go on for hundreds and hundreds of bases. You can have many, many of these repeats, and it increases the gene size just due to the increase in the number of these bases in here. There are a variety of diseases that are caused by these triplet repeats. Here's the repeat. Here's the CAG. This one is muscular atrophy here. Huntington's disease is another CAG. Fragile X is CGG, which we just looked at, and so on. Here's the normal number of repeats in these genes. And here's the number of repeats in a particular disease. And look at this. You could have 72,000 copies of CGG in your gene, which causes the Fragile X trait. So huge numbers of increases in trinucleotide repeats can cause these diseases. So let's look at a little more about this Fragile X and see what happens. The Fragile X syndrome, it's called FMR1, that's the gene, FMR1 gene. And here is a cartoon of the gene, and you can see that part of that gene is full of these different repeats. So this is the normal allele, and as it expands, it becomes bigger and bigger, and here's a really, really big one here. And so we have normal. This is known as the pre-mutation, so it's not quite fully mutated, but it's just got bigger. And here's the mutant allele that actually expresses the trait. In this case, if we have over 230 repeats, will give you the trait of Fragile X. Of course, it can go up to 72,000, but over 230 will give you the trait. So how do these repeats occur? I mean, what's going on as the gene replicates? It is to do with the DNA polymerase replicating the gene, and it tends to stutter, and it tends to increase the number of those repeats. Strangely enough, when we're looking at the maternal transmission, the number of repeats increase as she passes on that gene to the next generation. However, if you're looking at the paternal transmission, the number of repeats decreases or remains constant. And so there is definitely a gender effect here on how these number of repeats are passed on to the next generation. So Fragile X syndrome, here's a child with Fragile X, and if you look at this diagram of cartoons, it's called Fragile X because at the bottom of these X chromosomes, these are the female X chromosomes, you can actually see a change in DNA. It's visible to the microscope, and that's because of all these many, many, many CGG repeats. So heterozygous mothers, in other words, ones that have one bad chromosome, one good one, they tend to be phenotypically normal. Fragile X phenotype includes mental retardation, of course, but they seem to be phenotypically normal. Now, hemizygous males, that's the males with a Fragile X chromosome, you would think that everybody would display that, but only about 20 to 50% are normal, with about 50 to 70%, 50 to 80% phenotypically displaying the trait. And so in this case, you'll get a lot of Fragile X males rather than Fragile X females, because heterozygous females are normal, whereas hemizygous males are sometimes alright. That concludes Chapter 11 on mutations.